And whenever you're ready.
All right. So my presentation today will be about superbugs, which is going to be an analysis of the hyperresilience of advanced pathogens. So when we're talking about hyperresilience, we need to define what resilience actually is. So resilience can be defined as the ability to bounce back and recover from adversity or difficult situations. It's not a fixed trait, but it's rather a dynamic process that can be developed and strengthened over time. Resilience involves the development of specific skills and resources that enable individuals to navigate through these difficult situations and successfully navigate through them and continue to grow and flourish in the face of ongoing challenges. So what are superbugs? Superbugs are bacteria that have become resistant to multiple types of antibiotics, making them difficult or impossible to treat. These bacteria demonstrate resilience in several ways. So genetic adaptation, what does that mean? Superbugs have evolved mechanisms that enable them to adapt to their environment, including antibiotic exposure. These mechanisms include mutations into their DNA, transfer of resistance genes between bacteria, and the selection of resistant strains through overuse of antibiotics. Moreover, they can also form what's called a biofilm. These are complex communities of bacteria that adhere to different surfaces and are protected by a matrix of extracellular polymers. Biofilms provide a physical barrier that protects bacteria from antibiotics and the immune system. Moreover, we have efflux pumps. Superbugs have efflux pumps, which are transport proteins that pump antibiotics out of the bacterial cell before they can exert their antibacterial effect. This mechanism enables bacteria to avoid exposure to antibiotics and maintain their resistance. They also have enzymatic inactivation, where they can produce enzymes that block the antibiotics before they can reach their targets. Finally, they have horizontal gene transfer, where they can acquire resistance genes from other bacteria through horizontally transferring their genes between plasmids and bacteria. So why is this problem an issue? So the main factors are that there can be more prolific infections. With superbugs existing in the natural environment, the rate of bacterial infection can increase exponentially because these strains are resistant to the typical treatment methods that we have for them. Because of this, there's an increased difficulty to disinfect somebody who has been infected, which can lead to higher mortality rates and people dying as a result of bacterial infections, things like sepsis and so on. Moreover, there are heightened healthcare costs. Because of this, more people are staying in hospitals longer, and because of that, the previous treatments that we were employing are no longer working. So what happens if we ignore this issue? So as we can see with this graph, in the beginning towards 1981, the rate of antibiotic resistance was fairly low, right? Antibiotics were introduced, obviously, but their rate and the extent to which they were widespread was fairly low. Moreover, as we go on through time and into the 1990s and early 2001, we see that these MRSA, VRE, and QRP, which are strains of different bacteria, their rates of resistance to these antibiotics have increased. So if we keep ignoring this issue and we keep putting off dealing with these bacteria, the rate that these strains are growing and proliferating will keep increasing, and we'll have less means to deal with them in the future. So, how can we solve this problem? So the solution one is to enhance the status quo. So by enhancing the status quo, what we're doing is we're making the current situation of dealing with these bacteria a little bit more efficient. This can mean better stewardship programs, which are better training programs for healthcare staff and medical students that allow them to be exposed to these different types of resistant strains earlier and know their treatments a little bit better. Moreover, we can implement better medical advertisements. So there's a lot of direct-to-consumer advertising with medication, but there are also different types of medical advertising that can be used to educate people about these different types of bacteria and places that they can be spread more easily, like clubs or restaurants or other places where these bacteria might be a little bit more prolific in the environment. This goes hand in hand with public campaigns. So not only are advertisements going to be used, you can also campaign publicly about these types of infections to increase awareness for them. Something else that can be really done in the short term is to elevate the promotion of hygiene. So washing your hands, making sure you wear PPE when dealing with an infectious environment. These are all factors that enable individuals to combat the effects of bacterial exposure in the short term. Moreover, we can use, in the short term, the best approach that we currently have is strategic antibiotic use, right? So postponing antibiotic use if it's not as pertinent to the person's health, or if it's a lesser priority as opposed to other measures that we can use to combat those bacterial infections. The second solution would be to innovate for the future. So this would mean developing more efficient antibiotics, which is a very costly and inefficient process as it currently stands. But it stands to reason that with stronger antibiotics and with stronger procedural methods, we can combat the effect of bacteria much more smoothly, but with the drawback of increased costs and decreased efficiency. We can also implement a more controlled containment for these bacteria. So where individuals are in a high concentration area or a high impact area for these bacteria, maybe let's say a village that was infected with a bacteria, we can increase the containment or control the containment of bacteria within that region. Next, we can also establish international health policies. So this entails government cooperation to establish legislature that puts together processes and laws that basically that outline a guideline for how we should deal with people being in infected areas, and also the limitations that they can have with social gatherings and other areas where bacteria are prone to spread much more efficiently. Next, we can broaden research efforts. So this means increasing funding to those who are trying to research these bacteria further and trying to develop solutions that are that have lower costs and better outcomes. Moreover, we can lower experimental limitations. So this is a bit more of a controversial solution, but lowering experimental limitations would entail that we lower the bounds or we lower the parameters that we've set as constraints for the different experiments and research projects that aim to explore how these bacteria spread. Now, if we're using human subjects, obviously there are ethical concerns that this raises. But if we're using other, lab-grown mice or similar, just bacterial cultures in isolation, it's a little bit more ethical, but it also the effect of it can't be observed as easily. So as we discussed, enhancing the status quo increases investment needs, right? If we want to promote the effects of bacteria being spread and being a harm to society, we need better investment and better efforts that can actually cause change in the world for this solution to be implemented. There's also limited access to resources, equipment, and treatment facilities for the labs and hospitals that are trying to solve this issue, right? And as it currently stands, this is a fairly niche issue in the medical field, and they're not, superbugs aren't as widespread as we'd like them to be at this point, to be a forefront priority in a lot of medical research facilities. There's also constrained incentives, right? With limited funding, not everyone in the medical field wants to pursue a project that helps to eliminate these barriers. There could be an absence of public compliance. So when we're trying to emphasize hygiene and an increase in limiting gatherings, that might not always be observed in the areas that we're trying to promote it to or campaign for it. For innovating for the future, there's also a lack of funding and sufficient time. So with developing stronger antibiotics, as we mentioned before, there can be a lack of time and a lack of funding to develop those antibiotics. Just because we want them or we have the means to implement them doesn't mean that they're going to be implemented right away. Another issue is bacterial complexity. Typically, these strains of bacteria are much more complex than the average E. coli or similar strains. They have much higher rates of infection, and they can also mutate very rapidly, which makes it difficult to trace or combat them with one single drug in particular. There's also ethical and safety considerations, which we hinted upon before. So with ethical and safety considerations, research projects with these and labs with these bacteria need to have a very high level of containment in order to maintain safety. If any sort of slip-up happens, there can be a massive rate of infection and much larger issues such as a pandemic that we can't necessarily combat as efficiently. There's also the possibility for restricted international cooperation and regulatory barriers. So different countries and nations that have medical policies might have differing or contrasting viewpoints for how to deal with this issue. So some countries might prioritize research while other countries prioritize just emphasizing hygiene and doing the bare minimum. So it's vastly dependent on the economic and the sanitation status of each country that we're trying to deal with. So if we go back to each solution, we can address the implications that would result as a result of implementing them. So with better stewardship programs and increased medical advertisements, we can definitely have more educated medical residents and doctors that are better able to deal with when these infections arise. And with more public campaigns and increased hygiene, the public persona or the public attitude towards these highly infectious strains might be a little bit more familiar, and people might be a lot more open to implementing these strategies and to staying away from antibiotics and preventing the problem at the source. With innovating for the future, if it is accepted, we can definitely develop stronger antibiotics that might be a little bit more helpful in reducing the effect of superbugs and their effect. But we can also, with the implementation of more controlled containment, that is hard to implement, and it cannot always be upheld in the society, in the facilities that we need them to be implemented in, and there might be some backlash with more containment. Obviously, if we're trying to restrict the procedures and the freedoms of a specific medical institution, they might not always be amenable to that. We can also try to broaden research efforts, and by doing so, we can definitely learn more about superbugs and what their effects are. But by lowering experimental limitations, that can bring up more public dismay and disagreement for the movement and the cause, because not everyone is willing to be a lab rat, if you will, for these experiments, and not everyone wants to see the effect of what would happen if one of these strains was introduced on a much larger and wider scale. Thank you.
Okay. All right, so I have 2 questions for you. How did you select the strategies you used to gather information? Were they effective?
So when I was trying to gather information, I mainly focused on reputable medical sites such as NCBI and a lot of the other medical databases that have shed light on this issue. A lot of my research mainly came from past studies that are delving into the effects of superbugs and their spread. Could you repeat the second part of the question?
Sure. Were they effective?
So yes, they were effective to the extent that I was able to learn a lot more about what. So going back to this slide here, all these factors I was mainly, before the research, I wasn't too familiar with them, such as biofilm formation or efflux pumps, and the list goes on with these factors. But my research definitely helped in elucidating these components and allowing me to see the extent of how superbugs, the effect of superbugs and their resilience.
Okay, and then lastly, how did you use the conclusions and questions of others to advance your own research?
So a lot of the research that I was dealing with, they concluded especially with this graph right here, they were concluding that an increased resistance to these bacterial strains would result in higher infection rates. But I kind of tried to broaden that out a little bit and say, all right, if there are higher infection rates and higher resistance, that means that the public persona or the public perspective on these superbugs might be shifted a little bit more towards, hey, we need to deal with this issue, we need to combat this type of infection, especially with the COVID-19 pandemic. A lot of people became a lot more aware of what the effect of a highly infectious pathogen is and what it can do to a society. So I think other research is mainly centered, from what I found, on just specific strains and their effect in very isolated cultures. But I think we can broaden that out and just build up upon their ideas a little bit more that shifts to the public front.
All right. Thank you very much.
Thank you.